And our speaker is Thomas Tensfeldt. And Thomas is going to speak to us about OpenNCA, open source pharmacokinetic data repository, and non-compartmental analysis system. Thomas, take it away.
Good afternoon, everyone, and thank you to the conference organizers for inviting me to talk about the open source OpenNCA system at our Pharma 2020. I'm going to spend the next few minutes introducing you to a project developed at Pfizer that's a qualified industrial regulated platform leveraging the R programming language as the basis for analysis, visualization, summarization, and reporting through both direct user interaction as well as system-driven automation. I'll start, however, in briefly describing what NCA refers to. Non-compartmental pharmacokinetic analysis is a standard evaluation of PK concentration time data that are collected through clinical studies. There are other techniques of analysis. However, NCA provides a primary basis for exposure assessment in numerous clinical pharmacology trial designs, including bioequivalence, special populations like hepatic impairment, renal impairment, ADME studies, and so on and so forth. While the analysis approach would appear to be straightforward, generating pharmacokinetic parameters such as the maximum concentration over time, or the time of that maximal concentration, or even the analysis, the area under the curve, AUC, as well as others such as clearance and volume distribution, the information derived from these analyses are the basis for regulatory evaluation for marketing authorization and approval. OpenNCA, in the context of this, is a qualified desktop application and system leveraging R for much of its operation, and because of that is greatly extensible without additional system development. It comprises an R package for NCA analysis, as well as the framework delivering the repository, metadata, version control systems, file validation, audit logging, compute services, publishing, access controls, notification, authentication, etc., as well as user interaction with the data for analysis and reporting. It leverages Docker containers for R and RStudio for data transformation, visualization analysis, data summarization, and reporting in a traceable and reproducible manner. It's the open source replacement to Pfizer's internally developed web-based system, eNCA, that was S+ and WebLogic based, that went into production in 2007, but was replaced now on June 1st of this year with OpenNCA with the first production release. It remains under active development with a series of releases for OpenNCA planned through the end of this year and throughout 2021. I'll cover a little bit about OpenNCA's capabilities here, and then we'll go into the details and some visuals that will give you a better sense of what's possible. OpenNCA contains a user and system content library. In that, we store configuration content, we store basic standard code templates and reporting templates, and any other documentation that may be required. It's a general purpose repository, and it has capabilities both for a global system library that's controlled by user access roles, and a user library where the user can create his own content and use it in his analysis and reporting needs. There's an overall repository, the content data metadata repository, where we can do data loads to that repository. With the data loading mechanism, we'll show a little bit later that you can do field mapping, data merging, data validation. For the repository, there are authentication and data access controls, as well as navigation controls across the projects that are stored in the repository. There's a system for the traceability, and that's addressed in several different ways. There's traceability around how the data are modified throughout the system, who does it, when it was done, etc. And that's exposed mostly through the lineage map capabilities, and we'll show that in a little bit. There's dependency tracking on top of that that gives you a summarized view of that. There are workflow actions and an audit log, of course, as you would expect for a system that was intended for regulatory use. There's a search capability that allows us to search for content across the system. There's a non-compartmental PK analysis system, and that's part of the primary features of the system, That's an R-based package that we expose to the system, and an analysis module that the application provides us so that the user can interact with the data and the code very directly. There's a web-based data sharing facility that allows us to create a concept called reporting events, where we can collect data that's useful for a particular type of reporting need. Could be a clinical study report, could be for interim analyses, ad hoc analyses, interaction with data monitoring committees, and so on and so forth. With that, there's a publishing capability that's supported as well. I'll show that a little bit later. There's also an analysis, transformation, and reporting capability where we leverage not only R scripts, but we have deep integration with RStudio and Shiny apps that can be used. We have a standalone mode for the application so that you can work without all of the web services that are provided. And there's also the option for study metadata integration and patient information management system integration. And we leverage that at Pfizer, but it's kind of an optional capability. So we'll start with the traceability features. Here you can see that the OpenNCA application user interface presents with a series of frames that represent the navigation panel on the left, the lineage map or graph on the top right, and a summary panel on the lower bottom right. The navigation panel allows the user to flexibly search project and protocol metadata to select the protocol to focus on for a review and analysis. It leverages Elasticsearch of the repository and operational metadata to quickly identify project and protocols that match a simple text-based query. You can navigate directly by selecting from the list or enter a protocol ID or a search term to find the content and projects of interest. The lineage map represents the transitions of data from data load, on the left-hand side here, to data transformation and analysis and reporting items throughout the lineage map, with each entity node containing reference to the inputs and outputs, the scripts and logs, and any metadata required to reproduce the entity's results. The summary panel on the bottom provides the details of the content for each selected entity node from the map. For example, for a data transformation or reporting item or report node, there can be an input data set, an R script that generates the output data set, a script log where we capture the results of any running R script, and other metadata characterizing the results, date, time, who did it, etc., the version, and so on and so forth. In the case of more complex entity nodes, such as a non-compartmental analysis, like AN37410 or AN37394 here, or even a derived analysis like a DPRM 41649, the summary pane generates, the node generates multiple plot files or multiple inputs and outputs. The results will have all been represented in the summary panel below through a variety of subpanes. We'll turn next to the content and metadata repository capabilities and focus on, to start with, data loading. The system allows data loading currently through CSV ASCII files and Excel files, XLS and XLSX, and allows the data load capability. We're allowed to load multiple or arbitrary numbers of data sets into the system in a simultaneous go. The data load interface allows for field mapping, which I'm not showing here, but it's a visual field mapping which allows you to map to a file validation configuration standard if desired. All right, so with the input data set and the configuration standard, you can map to that standard through this interface. The mapping is not required. The system allows for selection of any data fields required for analysis or any other action, but it may be useful when you're trying to constrain the data to a particular format or standard. The data merge capabilities, which I'm also not showing directly here, support standard inner, left outer, right outer, and full outer join capabilities, as you might expect. Apologies for that. The file validation capability allows for extensive content and structure and metadata validation and verification on a field by field and across file, cross subject, and also against predefined subsets of fields as desired. This is optional, doesn't require it. File validation doesn't have to be enforced, and it can be configured to be restrictive if you need it to be. Let's move on to protocol registration, authentication, and data access controls. At Pfizer, we integrate the system with a project or a clinical study protocol registry database, as well as a patient information management system that we use at some of our clinical research units. The system allows for blinded control access as well as restricted control. We distinguish partly based on our SOPs, but that access control is pervasive through the system, and even from published external access to content. So all of those things are very tightly controlled, but it can be as open and as simple as need be for an individual's organization's needs. We'll move on to the user and global library. Here we see that the global library allows us to maintain system configurations, data validation configurations, data merge templates, standardized R transformation scripts, Shiny app scripts, reporting item, and report templates, other documentation, basically anything that you might expect that a global library or a library function might allow. It allows both the global library, it's basically a system library that's access controlled, and each user for the system has their own library where they can introduce their own content, their own scripts that they may want to use. They may want to demote a script from the global library, change it, and use it, and so on and so forth. So that's the basics of the user and global library. We'll switch now to data transformation, visualization, and reporting. For visualization, by this we mean we're able to use some standard Shiny applications. You can use a default one that is accessible immediately when you select this option through the interface. So you select your data or your analysis and trigger the visualization, and it starts immediately by running the Shiny app in a subframe. This is a simple demonstration of a spaghetti plot that we use for demonstrating the capability. But these Shiny apps can be as complex or as simple as you need them to be. We can generate plots, tables, reports, and so on and so forth. This particular use in this part of the application is basically to get a view of the data, and this could have any number of different types of plots and summaries to support that. This is what we have at the moment, but we're working on more enhanced apps going forward. The application uses R extensively. We use it here. You can see one of the data transformation compute nodes. Here we're creating or updating a data transformation where we bring input data that you can see in a preview pane down here, along with controls to select different columns, filter the data in a variety of ways. The script, this original script that generated this exists here. It's displayed along with any metadata associated with the data transformation. The compute pane, when I triggered it, loaded a standard script. I can select a script from the library. I can type a script in, or I could even run it not as a script, but run it from an RStudio IDE, which I'll show in a moment. Here we can select any number of inputs to the data transformation. And we allow, as I mentioned, RStudio sessions, Shiny app development here, units conversion. I can do the Shiny app development through the RStudio session. I can do it here in kind of a crude way, but also we also support a standard units conversion for input concentration data sets, as well as NCA analysis output parameter data sets. Let's move on to the next. The RStudio IDE integration, that's accessible. So rather than working with a simple script in a simple edit window, you have all the power and features of the RStudio integrated development environment. And here you can see we have a simple script that generates a couple of plots that'll be used. And once they're saved and closed, it'll be returned back to the repository so that it can be incorporated as reporting items for reports, or just to explore and evaluate in the midst of data analysis. The system also permits our markdown and LaTeX report generation. And it does this through a capability that allows us to share the results of those of those reports as well. The example here illustrates a system-generated audit trail that captures all the content and metadata for quality control, quality assurance, and traceability purposes for a reporting event that includes input concentration and output parameter data. You can see on the right here, we have input data transformation, basically just the input concentration data. And then we have, from the analysis, a primary parameter data set, which are the results of the non-compartmental analysis. Both of these are published for a reporting event that we've identified as a clinical study report type. These are configurable. We have a variety of options in here for Pfizer's use of the system, but they can be configured to reflect anything. Once published, the configurable URL is used to retrieve the content based on authorization controls, and that URL can be manipulated by different options, which allows the content delivery to be configured in different ways. The back end of all of that is driven by R on the server. We recognize that while the system provides a mechanism for using R scripts and RStudio and Shiny and things of that nature, but we know that many pharmacokineticists are not programmers by training, nor by inclination. And because of this, we've introduced a mechanism that we call compute palettes that can automate script execution for the analyst. They really don't need to know any R programming if they don't want to learn that, or cannot learn that, or frankly don't need to learn it. The system can be configured with options so that we have standardized scripts, standardized assessments, standardized reports, Shiny apps, and the like. And in accessing these, you can choose to direct the script in with different parameters, so that the scripts can behave in different ways, or actually trigger a Shiny app to do everything that a standard script might do. So it can be done in a variety of different ways. And as I mentioned, you really don't need to use R programming to drive the system, but certainly you can, obviously. Let's turn now to non-compartmental analysis. I'll give you a little bit of an overview of how the NCA system works with this. The computation engine, as we call it, for the non-compartmental analysis that OpenNCA provides is defined through a series of models. We've defined these models as extravascular dosing, 2 intravenous dosing models for bolus and infusion administration, and as a urinary model that allows for interval sampling and typically used for urinary data. All models incorporate single and steady state parameter computation and standard parameters for clinical PK data evaluation. They allow for dose and body size normalized parameters, as well as automation for metabolite ratio parameters. And in addition, the steady state model rather uniquely allows for multiple dosing intervals within a sampling profile. And in fact, those dosing intervals don't have to be even. They can be odd intervals like 10 hours and 14 hours, or anything you need them to be, actually. This R-based package is based on a legacy S+ computation engine code that we ran at Pfizer for almost 14 years through the legacy system eNCA, and was used in over 1,000 studies with hundreds of regulatory submissions that were supported through it. And it's been fully qualified and gone through software development life cycles to ensure that it's in good regulatory shape. The computation engine package, of course, because it's an R package, can be used with any pharmacometric workflow. So if you want to use it in the context of evaluation of PopPK as a qualified system of NCA computation, you certainly can. To give a sense of how the system allows the user to do the NCA analysis, We can see here the analysis module that's accessible through the application. Once you trigger this, you can select your model, set all the configuration variables. And I'm not going to go into the details, but here you can choose to do virtually anything you can imagine. It's all driven from a template that we use that we call the model configuration template. And that's used to select the data, which could be standardized or changed from the data itself. We can generate partial areas, and that's done in a very flexible way as well. You can generate any number of partial areas, and it implements interpolation and extrapolation. If the extent, the range of the partial area that you selected is not part of the nominal actual time. The quote-unquote KEL module is where we actually do the analysis for NCA. And here you can select data for the terminal phase. You can choose points. You don't have to select data for the terminal phase. In a steady state analysis, you may not do that, but you can if you need to. You can select to exclude points directly from the chart. I didn't illustrate this here. You can view the results of this and the flags that are used for controlling incorporation of data in the terminal elimination phase and or area under the curve computations, or exclude a point from all computations if it appears to be aberrant, or even flag the data for incorporation or exclusion from summary reporting downstream. We can show the data in table format. We can switch, of course, back and forth between semi-log and linear views of the data. We can see estimated concentrations. If we're using a bolus model, you may want to see C0 in the application and how it's computed. We can include or exclude entire subjects or profiles, and so on and so forth. There's a lot of capabilities here, and there's new capabilities that I haven't even sort of mentioned here in the latest version of the system. We can drive the analysis of steady state data, both from incorporation of data, tau and told values that define the dosing intervals directly in the data itself, or you can also set those values on a profile by profile basis directly in the analysis module. And these will override what comes through in the data. So if you see something and need to adjust it, you can do that directly here in the analysis module. Let's move on to search capabilities briefly. With search, we're able to search for any study, any program, any entity within the system. At current, we have a limited subset of different status metadata that we can search for. We can search across different date ranges. We can do full text searching, which is quite powerful. We can even build a flexible data search where you can put in arbitrary expressions, for example, dose greater than 500 milligrams and gender equal to female, for example. So you can search for all of the data elements in the system, and the search results at the moment come back as links where you can navigate directly back to the protocol and lineage map for which the data appears. Eventually, we'll have this set up so that we can create aggregated summaries of data and use those to create cross-study evaluations within the system. We don't have that quite yet, but that's where we're headed with the search capabilities. So we'll move on to open source and where we are at the moment. Pfizer, conceived OpenNCA from the beginning as to be an open source project, but really to foster that, we've joined with, in collaboration with the Pharmaceutical Open Source Software Consortium, which you may not have heard of, but was established in 2018 in collaboration with 11 sizable pharma companies. The vision for the consortium was to promote innovation and collaboration through the development of open source software for clinical pharmacology and pharmacometric applications to serve the community, standardize common data elements and common elements that we use in our discipline, strengthen the discipline, and ultimately bring new medicines to patients. That's the vision, that we can collaborate together in a non-competitive space to produce things that we can all use to produce new medicines. The consortium is currently in its feasibility phase. It's been there for a bit. It's a current status where we've established the formation of the consortium. We've defined the requirements for an NCA software product and have initiated the public solicitation for interest and information to deliver the solution to a variety of vendors. We haven't moved into a development phase yet, where we would go through vendor selections, requirements finalization, and initiation of development. We would commence that once the decision to proceed has been reached and continue until the first project, the NCA software application, is delivered. Apologies for that. OpenNCA is being offered by Pfizer to bootstrap the consortium NCA project. And that status, apologies, is in evaluation currently. So we presented this as an option to initiate the project and use it as a basis for future development efforts from the consortium. Ultimately, the goal is to broaden access, foster collaboration through the industry, but also eventually through academia, regulatory agencies, CROs, etc. So in summary, OpenNCA is a novel open source NCA software solution and environment. It's fully extensible without redeveloping the software from scratch. It qualifies for regulatory use with a full slate of software development, testing, controls, and documentation. It's, I believe, a good example of a qualified industrial application of the R programming language, and it's a basis for industrial collaboration through open source software development and innovation for the clinical pharmacology and pharmacometrics disciplines. Thanks for listening, and if any of this interests you, please reach out to me, let me know. We're quite eager to work with the consortium, as well as others in the industry and beyond. We believe this system has great possibility for collaboration with the community and helping to advance the state of the art. Thank you, and good afternoon.